Hi, and welcome to Teenu Thomas. Today, we are going to learn about bacterial cell. What are bacteria? They are single-celled microorganisms. We cannot see them through our naked eye, but they are present everywhere. Very interestingly, the smell of rain is also because of bacteria called actinomycetes. The bacteria are present inside our body, outside our body, or on the surface of a table. They are also in our diet. This is a bacterial cell. We are going to see them in detail. The first one is cell wall. Cell wall is the external structure in a cell. It's very rigid and gives shape to the cell. Its main function is to prevent the cell from expanding and eventually bursting due to the water uptake. Cell wall constitutes a significant portion of the dry weight of the cell, and it is essential for bacterial growth and division. The cell wall cannot be seen by direct light microscope and does not stain with simple stains. Staining is a procedure we follow in microbiology lab to highlight the cells or a microorganism under microscope. Only through electron microscope or certain other procedures we could see cell wall. Next one is outer membrane. Outer membrane is found only in gram-negative bacteria. Gram comes from the name of bacteriologist Hans Christian Gram, and we refer Gram by the capital letter G. In 1884, Gram devised a test to identify whether or not a bacteria had a peptidoglycan, which is actually a mesh-like layer of sugars and amino acids. In his test, what he did was he introduced a dye to the bacteria. If the bacteria had a thick peptidoglycan cell wall, it absorbed the dye and turned purple color. It tested positive for peptidoglycan. If it did not turn purple, it tested negative for peptidoglycan, meaning its peptidoglycan layer was thin. As this method was adopted, the resulting categories were called gram-positive and gram-negative. This method of gram staining is still a widely used standard procedure in microbiology. Next one is lipopolysaccharide. The lipopolysaccharide or LPS present on the cell walls of gram-negative bacteria account for their endotoxic activity and antigen specificity. Endotoxin and exotoxins are present in bacteria. Endotoxins are actually present inside the bacteria and will be released only during the bacteria dies. But exotoxins will be released outside environment during the bacterial conjugation or during the metabolism. They are also known as lipoglycans and endotoxins. They consist of lipids and polysaccharide, hence the name lipopolysaccharide. The main function is the structural integrity of bacteria and protecting them from chemical attack. Next one is cytoplasmic membrane. The cytoplasmic membrane is present immediately beneath the cell wall, found in both gram-positive and negative bacteria, and it is a thin layer lining the inner surface of the cell wall and separating it from the cytoplasm. It acts as a semipermeable membrane, controlling the flow of metabolites to and from the protoplasm. Protoplasm is called as a living substance of the cell, and it surrounds the cell membrane, the nucleus, and the cytoplasm. Cytoplasm is one of the constituents of the protoplasm. It is a jelly-like fluid and contains water, metabolites, waste, proteins, and food reserves. Next is the cytoplasm. Cytoplasm is a colloidal system which contains both organic and inorganic solutes, and the main composition of cytoplasm is water, and it also contains salts and proteins. They are rich in ribosomes, DNA, and fluid. DNA is circular and haploid, and also you can see in the picture they are highly coiled with intermixed polyamines and support proteins. Ribosomes are the centers of protein synthesis. In the picture, you can see ribosomes as a circles. Next is the mesosomes. They are vesicular convoluted tubules formed by invagination of plasma membrane into the cytoplasm. In cell biology, a vesicle is a structure within or outside a cell consisting of liquid or cytoplasm enclosed by a lipid bilayer. They are the principal sites of respiratory enzymes like cytochrome oxidase, ATP synthase and all. Next is the nucleus. As we know, bacteria are prokaryote, so there is no distinct nucleus and has no nuclear membrane or nucleolus in it. Next is the capsule. Capsule is the outermost layer or the extracellular part of a bacteria. It's a condensed, well-defined layer closely surrounding the cell. They are usually polysaccharide. And if the polysaccharide envelops the whole bacterium, it is capsule, and the production depends on the growth condition. When it forms a loose meshwork of fibrils, as you could see in the picture, extending outward from the cell, they are described as glycocalyx. And when mass of the polymer that formed appear to be totally detached from the cell, and if the cells are seen entrapped in it, are described as a slime layer. Flagella. They are the long hair-like helical filaments extending from cytoplasm membrane to exterior of the cell. Flagellin is highly antigenic and functions in cell motility. The location of the flagella depends on bacterial species, as pole are situated at one or both ends, which swims in back and forth fashion and lateral along the sides. Next is the pili or fimbriae, which is also similar to flagella. They are hair-like proteinaceous structures that extends from the cell membrane to the external environment. They are thinner, short, and more numerous than flagella, but they do not function in motility. Pili allow for the transfer of DNA between bacteria in the process of bacterial conjugation. So we had seen different areas in a bacterial cell. They were cell wall, outer membrane, lipopolysaccharide, cytoplasmic membrane, cytoplasm, ribosomes, mesosomes, nucleus, capsule, flagella, and pili. If you want to read more about this, just go through these books. Thank you.